Okay, now let's move on to the next most common family that we find on bodies. These are the houseflies, or the Muscidae. So muscids are commonly known as houseflies or filth flies, dung flies, sweat flies. I think you can sort of see all the commonalities here. And these will include significant blood-feeding parasites, vectors of disease agents, and just really freaking annoying adults. Anytime you have one of those flies that's in your house, it's just all in your face, it's a freaking muscid. I hate muscids. So now this family is what we call synanthropic. So a synanthropic organism means it's a species that exploit foods and habitats created by agriculture and other human activities. So these are flies that follow us around. That's why they're in our garbage, in our homes, in our faces, all of that sort of stuff. Now we group together the Muscidae into common name groups, basically based on their affinity or their habitats. So filth flies, they frequently visit a variety of organic substrates, including latrines, household garbage, manure soiled animal bedding, and carrion. So these are ones that we do see ovipositing on dead bodies quite a bit. Dung flies exclusively occur on droppings, especially cattle droppings, but we do see some of these dung flies in humans every once in a while. Sweat flies have adults that feed persistently on perspiration. So these are the ones that land on your faces, land on the animals, and try to feed on sweat. So we get a lot of these really obnoxious flies showing up, but they are decomposers. They do show up in garbage. Now there's approximately 4,200 species in this family, 700 of which occur in North America. There's only a few of the genera, though, that are important when it comes to pest status or when it comes to bodies. 5 of the species that we're looking at are introduced from the Old World. Because of their synanthropic capabilities, they have come over with us when we came over to North America. And like all Diptera, muscids are holometabolists. They have an egg, a larvae, and a pupal stage. They then turn into their adults, which just start the thing all over again. They are anontogynous. Remember what that means? They require a protein meal to produce eggs, and so then they have to find a suitable oviposition site in order to lay their larvae down, lay their eggs down. Those larvae need to feed. They mimic a lot of the same things that Calophoridae do. They look like Calophoridae, their eggs look like Calophoridae, the larvae can be kind of hard to tell apart, the pupae are hard to tell apart. So they're very, very closely related, and this can cause some issues if you don't know what you're looking at. So the eggs are similar to Calophoridae eggs, and depending on the species, they're either going to be laid down singly or laid down in groups. Now, the larvae are creamy in color. Okay, so they are known as maggots, but they're this sort of yellowy cream coloration. This is the way that I tell them apart from Calophoridae, like in a maggot mass, is because the Calophoridae tend to be bright white, unless you're talking about Chrysomyia rufaces, but the Muscidae tend to be this sort of yellow creamish color, and they also look much smoother. They don't have nearly as obvious ridges around each segment. So it's easy to sort of compare on sight the difference between the larvae. However, you always want to double check this when you get back into, say, the lab to make sure you actually know what you're dealing with here. So the larvae, like the Calophoridae, they have 3 larval instars before they reach adulthood. They've got their mouth hooks, they've got their posterior spiracles, all of that sort of stuff. The big thing with the posterior spiracles is instead of being straight like we saw in both the Calophoridae and the Sarcophagidae, the posterior spiracles in Muscidae are sinuous, so they're S-shaped or squiggly shaped. So they look very, very different. The head of the larvae is greatly reduced. It doesn't have any eyes. It has very minute antennae, very, very tiny, and they're supported internally by a sclerotized cephalopharyngeal skeleton. So this is a very complex structure. It's partially visible through the integument of live larvae. You saw this a little bit with the close-ups of the Calliphoridae, but it is very important for identification in a lot of these species. Now, the paired mouth hooks are present. They can be extended and retracted from the oral cavity, and they will help in crawling and burrowing and in tearing into food. Most of the larvae occur in very wet substrate, so decomposing material, fecal material, dead bodies, that sort of thing. So their paired spiracles on their prothorax and on their abdomen allow them to breathe. And the characteristics of the spiracles, they're the most useful for determining the species and the instar of the muscid larvae. The prothoracic spiracles, so these prothoracic spiracles are just on that first thoracic region in the larvae, so they're right there. They are absent in first instars and present in the second and third. And then the shapes of the spiracular openings in the and the number in the spiracular openings in the posterior spiracles are the same as others. So the first and second instars have 2 spiracular slits, while the third instar has 3. And each spiracular plate consists of a peritreme, a ridge around the outside or the perimeter of the spherical, and they have 2 or 3 slits for gas exchange. Now, the peritreme shape, position, and opening size are used for identification, and those squiggly slits help you ID down to Calliphoridae rather than Muscidae or down to Muscidae instead of Calliphoridae or Sarcophagidae. The adults are 4 to 12 millimeters long, so they're a little smaller than the sarcs. Their wings are longer than the abdomen, so that's probably the easiest thing to see. And they tend to be brownish gray to black, and oftentimes they will have vitti on their thoracic region. Okay, so they've got those dark vitti, and they will have these dark splotches on the abdomen. It's not tessellated like we see in Sarcophagidae, but it can be splotchy. Now, the vitti, they tend to have 4 vitti on their thorax rather than 3 like we see in sarcs or in Calliphoridae. Now, the head is 3 ocelli and a prominent pair of compound eyes, and just like the Calliphoridae, you can tell the difference between males and females by the eyes. The females, they're very, very far apart. In males, they are touching. Now, most muscids are oviparous, so they will lay down eggs. They deposit fertilized eggs in the environment before they hatch. So unlike Sarcophagidae, these again lay eggs like the Calliphoridae. Now, oviposition substrates vary among the different species. Filth flies oviposit on organic debris. They like these really wet organic debris and wet enough to support aerobic microbial fermentation. So they like things like feces, they like garbage, they like algae, long clippings, food waste, all that gross stuff. Where you've got confined livestock and poultry, they will lay their eggs in manure, soiled beddings, wet feeds, rotting stuff, that sort of thing. Dung flies and sweat flies are a little bit more choosy. Dung flies will oviposit on or in cattle dung pats within minutes to a day or so after the animal defecates, while sweat flies will oviposit in plant litter and decomposing dung in grasslands and forests, and then the adults will go after the sweat. Now, larvae of the muscids will burrow. They will feed and develop in their respective ovipositional substrates. The larvae feeding differs among the species, even among the instars of the same species. Most muscids are saprophages. They feed on bacteria, on yeasts, and organic particles in the decomposing matter. The third instar of some of these species, however, are facultative predators. So they're a lot like the Chrysomyrufosces there. They mature as saprophages, so they feed on decomposing matter, but they're going to switch to predation. They're going to consume other soft-bodied insects if they are available. So muscids can act as biological control of other maggots in the area, but that means you have to put up with muscids. So it's really a crapshoot right there. Now, once they're mature, the third instar larvae will cease feeding like every other prepupal larvae. They'll empty their alimentary tract by defecating, and then they're going to wander away. So they're going to move away from their feeding substrate right before they pupate. Now, newly emerged adults of muscids have very little stored energy and nutrients. They don't do a lot. They need to find water and salts and carbohydrates and proteins to survive. The non-biting species, those with sponging mouth parts, will obtain sugars from things like nectar and honeydew. Remember, honeydew is the urine or the poop from Hemiptera. So the biting flies, there are a few flies that will feed on blood, and they get the bulk of their food from blood or from serum, from saliva, from mucus, from lacrimal secretions. So they can land on living animals and feed on those. The activities and locations of adult muscid flies vary with the time of day. Most are active during daylight hours, and almost all are inactive at night. They will fly, they will locate hosts, they will feed, they will mate, and they will oviposit all during the day. Sight and olfaction are used to locate hosts and oviposition sites, just like we saw with the Calliphoridae. Most muscids, though, do not enter houses, but a few notable species do, specifically the housefly. So these are the ones that we can see ovipositing on rotten dead things that are indoors. So we do have a couple of muscid species, these houseflies, that will move inside. Now, the flies find daytime resting sites according to their needs for thermoregulation. They rest in sunny sites when the air temperature is below about 20 degrees Celsius, and in shady sites when it exceeds about 30 degrees Celsius. So they like it pretty much like we do. You know, they like the 70s and the 80s Fahrenheit. If it's too hot, they go and sit in the shade. If it's too cold, they go and sit in the sun in order to warm up. So you'll see them resting during the day in those areas. Now, the flight range of muscids is extensive. They can fly very, very far. I mean, detectable numbers have been collected more than 5 kilometers from their points of origin, which is really far for these little tiny flies. And some of the larger species, like stable flies, can appear on beaches 10 or more miles downwind from where they bred. So they can ride wind currents. They can move really, really far. So they're very strong flyers, which means that they're going to be able to find decomposing material in a lot of new places. And this helps them with that synanthropic tendencies, where they like to follow humans around. Now, the seasonal patterns of muscids vary among species. It varies among the years, it varies among the location, so everything is sort of up in the air with these things. In areas that have very, very cold winters, the population growth outside buildings is restricted to a distinct breeding season. The warmer months of spring, summer, and autumn are when we see the houseflies, and the population is going to grow to a single peak in early autumn, and then they're going to die off during winter. The notable exception to this is the housefly. They breed continuously because they go indoors, they move into heated buildings. In warmer climates, breeding season for most species is much longer, and depending on how close you are to the equator or how warm it is, it may be continuous or year-round. Most muscid flies of both medical and forensic importance are what we call multivoltine. Multivoltine means they develop through 2 or more generations per breeding season. And these generations usually overlap. So you'll see eggs, larvae, pupae, and adults of all ages present simultaneously throughout most of the breeding season. So in these warm months of the year, you're just going to see all stages of development all over the place. And population growth within a breeding season is influenced by the availability of breeding media, so all the decomposing material, places for them to mate. It's governed by the weather, how warm it is, and the individual reproduction rate of each one of these species. So some will produce a lot of eggs, some won't. It just depends on what species we're dealing with. If it gets cold, muscids will overwinter in different ways. So in those that are lucky enough to live in frost-free southern regions, they're just going to breed continuously, maybe a little slower in the winter, but for the most part, they're going to keep going. They're going to retreat to semi-heated substrates associated with humans and livestock during the coldest areas, and we see this happen a lot also in the northern regions. So they will move into heated barns. They'll move into outbuildings. They'll move into places where there is climate control. Other species, though, will experience diapause. So diapause is a state of developmental arrest associated with tolerance for freezing. Flies diapause most commonly in the adult or the pupal stages. These are the hardiest stages. So when it gets super, super cold, we'll find these muscids just shutting down their body and sleeping it off, basically, until it warms up. All right, so that's the introduction to the Muscidae. Up next, we're going to look at a few of the forensically important species. Let me know if you have any questions.